I met Dr. Telugu it's probably been almost a year ago now. We were interested, the CRF was interested in looking for a large animal model in which we could study choroideremia. For 2 decades now, there's been small rodent-sized models and even models at the molecular or cellular level, but we've never had a large animal model. And that's how our paths crossed. And Dr. Telugu is the founding member, president, and CEO of ReOVAte Biosciences. And he's also an associate professor at the University of Missouri, Columbia. And his laboratory is actively engaged in developing porcine models, pig models, of human diseases. Specifically, he's looked at diabetes, cardiovascular disease, and obesity. And he's looking at ways to develop technologies and translational research where a larger model would be more appropriate than a small animal model. And with that, we'll listen to this presentation by Dr. Telugu. Thank you very much.
Again, many thanks for the CRF Choroideremia Research Foundation for first of all funding this program in our lab, and secondly for invitation. It's one thing to be doing this in the lab and understanding what the disease is, but it's another thing to come and actually meet with the patients and family. So I can promise you that I'm going back with renewed purpose, and we'll try to do whatever we can to accelerate the pipeline. So with that, I have to declare a conflict of interest, as Dr. Jess Thompson mentioned. I actually founded a startup company. I'm a faculty member at University of Missouri, and the reason to start the company is actually for the precise reason. I mean, it's one thing to do at the university where things go at a snail pace, and at a company we can put it on a front burner and we can go at an accelerated rate. And another thing is it's advantageous for us to be, and it's easy for us to put together proposals and work with the pharmaceutical companies. And my hope is that once we have done this preliminary validation, we would engage the pharma to then take this to the bedside. So, from bench to bedside. So that's the hope. As I said, the goal of the company is to generate these custom models where pig is an ideal model for studying and also for developing therapeutics. And again, we work with both academic institutes as well as pharmaceutical companies. The core of what we do in the lab as well as at the company is to use genetic engineering, and we use that to address critical priorities in both ag and medicine. I'm not going to go into too much into ag. We have several projects that are funded by Bill and Melinda Gates Foundation to develop animal models, animals that are resistant to diseases, for example. We are the first company that enabled a group to develop pigs that are resistant to a disease called PRRS that's actually going through FDA right now for approval. So we actually have some knowledge on how to progress these to FDA pipeline for commercialization as well. On the biomedical side, a predominant focus has been on using these models and leveraging these models for biomedical applications, predominantly for disease modeling. As Dr. Thompson was mentioning, your model, your treatment is as good as your model. So, and I'll explain why we are going through this pipeline in a minute. And we also work predominantly with regenerative medicine applications. So CHM falls into that paradigm where we're trying to use stem cells, including the patient-derived stem cells, to eventually come with a transplantable, therapy for the patients. And just before I go further into the discussion about the pig models, I just want to mention it that I have no bias against animal models. I'm a veterinarian by training. So I'm species agnostic. I ask the question and go for a model that best answers that question. Okay. For these eye models, I believe the one that's going to deliver to a promised land is going to be the pig model. Okay. That's my bias there. And we also work on One Health applications. So studying flu, for example, in pig would have applications for flu in humans. So that's the idea. So I've broken up the talk into three major points here. One is, first of all, why we're using a pig model, right? The rationale for using the pig model. That's going to be the bulk of the talk. And I'm going to share a little bit of scientific data slides. Few of them, I know the audience is not scientific, so largely scientific. So I'm going to share you some progress and a way forward. So the fundamental question is, why pig? So the core argument is that the mouse model cannot fulfill the complete spectrum of biomedical research needs. So we have a tendency to pigeonhole everything through a mouse model. Again, no offense to anybody who's working with mouse models. As I said, I work with mouse models in my lab too. And it may have to do with the fact that the stem cells were first discovered almost 4 decades ago in mouse. And the guys who discovered it actually got the Nobel Prize. You know the second model he tried to make stem cells from? It's actually from pigs. And he failed. So that set us field back for almost 40 years. So you can see how the genetic engineering is more facile, easy to do in the mouse models. So that's why that model is at the forefront of all discoveries. That has been the major rationale. Okay? But that said, many treatments that we are developing based on mouse models are actually failing to advance in many of these clinical trials. And those drugs that we actually do, in fact, develop them are also failing miserably. So it's not only accruing a lot of cost, but also lost time. So here are some representative examples. So as seen on the top, there are almost like 100 drugs that are advanced stages of clinical trials that have failed. And the basic foundational science was done in mice. And that number is very conservative. Now, most of these actually don't even go through phase 2 of the clinical trials to begin with. So this could be in thousands of trials that are failing miserably, and we keep repeating the same mistake. So hopefully that's the driving point here, right? We argue that the pig model is an ideal bridge model. So let's say you confirm the findings in mice. And I don't know if many of the audience know that the research in monkeys or non-human primates, as we call it, is becoming incredibly difficult in the United States, and arguably so. So we may end up with a scenario where, you can see how the disease from mouse, the drug from mouse may enter into humans right out the gate. So you're actually putting patients at an incredible risk as well. So we argue that having a bridge model that would probably give very good preclinical safety data set might be a good way to go move this forward. And we argue that pig would be an ideal bridge model in this case. We do have a pharmaceutical company that's actually working with a pig model for a gene therapy using cardiovascular. So we're enabling those studies as well. So I know that there is greater reckoning about using this model in the field. And it's also a dual purpose model, meaning that you develop a treatment in this, it can go to humans, but also an immediate veterinary application is in place. So you develop a drug and you can go to veterinary applications really quickly, and that will develop a safety trial that will enable FDA to say, oh, now go into patients. Because now we have efficacy trials in your pig, we have efficacy trials in other veterinary patients, clinical trials. So we're good to go. Go forward and try them in the patients. So you can see how this could be an ideal model to consider. And relevant to this discussion, pig has a similar size. And we did genome sequencing, and we have confirmation now. Men are almost pigs at this point. So 99%, we are there. So we're very similar in size, we're very similar in anatomy, we're very similar in physiology. And more importantly, physicians do work quite a bit with the pig model. So as they're going through their surgical internship, they actually do surgical practices in the pig model. So most of the physicians are very familiar with working with the pig models. And the reason is that their size and the surgical approaches that you use are very similar to in patients. There are similar routes of delivery. So in the mouse, for example, there are only 2 or 3 ways you can deliver a drug. Tail vein and a few more stuff. So veins are, it's difficult to find a blood vessel to inject them. But in humans, pig, it's same. So you can basically say, I want to deliver this through this vein in patients. I can do the same thing in the pigs. So route of delivery is the same. We could also do longitudinal studies, and we can continuously do biopsies. We can do the tissue sampling. So it allows us to get more robust safety data set from these animals. And relevant to this discussion, the organ size is quite similar to that of humans. And this is very, very important, especially if you're trying to develop cell therapies and gene therapies where the dosing is important. Let's just say I come tomorrow and say, hi, I treated this disease in a mouse model. Well, by the way, the eye size is this. I'm going to guess what is going to work in you. That's not a good discussion to have with the patient, right? So our argument is that if I can cure a disease in a pig eye, like say CHM, because that's the topic here, I now have a starting titer, starting dose. I don't have to do guesswork. I know it'll work in humans, right? And lastly, this may be important. What are the long-term outcomes of this? Immediately we know treatment works, but what are the long-term outcomes? Well, mice lives for a month, for a year. After that, it's dead, right? But humans live for a very long time, hopefully, right? So this model allows us to look at long-term outcomes of these therapies, right? And among
recording in progress.
Say, oh, I like goats. Why don't you work goats, right? Or something else. I think pig offers several advantages for what we're trying to do, other than what I already mentioned from a genetic engineering standpoint. So pig actually is a litter-bearing animal, meaning that if I generate something, I can make a lot of them, which becomes important for clinical trials. Right? If I work with the cattle, I got one calf in 1 year. That's not a good starting point for me, right? And the gestation interval is also solved. Among all the large animal models, pigs are the only ones that have the shortest of the gestation interval. It's about 3 months, 3 weeks, and 3 days, right? Less than 4 months. So in less than 4 months, I have an ability to have more than 15 piglets from one pregnancy, meaning that I can easily scale them up. And they come to reproductive age in a very short time. So we can have a breeding herd of CHM pigs that we can then use for developing therapies, safety studies and so forth. So that said, we know that this is a good model. We know that for a very long term. It's a running joke at the National Institute of Health, actually. The very first grant under an R01 mechanism, which is the major grant that is funded to the researchers, was actually to a person studying pig model. But if you look at the models that are funded now, and more than 90%, I mean, easily, let me say, less than 1% of them are getting funded to pigs. So I asked the question, okay, it's important. It's a very good model. Why are we not getting a lot of funding? Problem is that the genetic engineering, as I said, is not very easy in these animal models up until recently. Something as simple as just injecting a DNA into an egg is kind of difficult. So one in the left is a mouse. You can see it's clear. You can see where the nucleus is. You know where to deliver. You can deliver that. On the right is the one from the pig. It's a black box. You don't know where you're going with this in the injection needle, right? It's very difficult to do these trials. And the second thing is a longer generation interval. So if I'm making a mouse model, you don't need to go into a lot of these details, but it takes approximately 1 year for me to make a mouse model that has the right mutations, right? If I go with the traditional breeding route, it used to take 3 years for us. So when I was doing PhD at the University of Missouri, and the entire PhD thesis could be develop a pig model. So that would be 4 years of research just developing a pig model. That's not a good career path for most of the people, right? So those were the difficulties back in the day. And that's why we did not hear more about pig until recently. Well, until here now. But then there is a game changer. You heard something about genome editing. Most of you heard about it, right? Genome editing is now a common name. It's coming to our vernaculars right now. So Jennifer Doudna, Emmanuel Charpentier, they got a Nobel Prize last year for developing this technology. That's a game changer. So we are the early adapters of these technologies in our lab. So the discovery was made in 2013. We were the first lab to try it in the pigs. And they work phenomenally. So all you have to do is get some embryos, inject them, and you can do whatever modification you want to do in the animal model. So I want to create a modification that some of the CHM patients have. All I have to do is find the eggs, inject those reagents, I can make a pig model with that mutation. Right? So I think the game changer is that. So I'm not going to go into all of these details here because it's not relevant for you. So I'm going to tell you the model we use in the lab. We use a method called SCNT or cloning. So most of you heard about Dolly the sheep, right? So it's the same process we use in the lab. So we start with cells. We make the genetic modification. Let's say in this case, we want to make a pig that is green, just example, all right? So we make the genetic modification. We start with oocytes. We take the nucleus out. We put this cell into the embryo, do the embryo transfer, and you should get the piglets. The advantage here is whatever modification you made to the cells beforehand, the piglets will have the same modification. So there is not going to be any surprises after end. And everything goes well. You have the GFP pig, right? So it's a preordained modification, so there are no surprises. So again, advantages are, you can make a lot of these animals that are clones that are identical of each other. So you have more biological replicates that are already built into it. So you can have some patients that get the treatment, some that does not get the treatment, but they're all within the same genetic modification. So it's easy to study the effect of modifications in these animal patients, right? So with this paradigm, I'm going to revisit that original problem I mentioned, right? In the case of mice, it used to take us 1 year to make the modification. So that's why everybody just went with mouse models. But with this new technology that we have in the lab, we can actually make these pig models in about 6 months now. For the first time in a very, very long time, we can make these models faster than a mouse model, right? It's doable. So now the question is, if you know this is a good model, why, and you have choice between a mouse and a pig, which one do you want to go, right? So that's what the question we're asking. Like, it's no longer a limitation for us. We can make these models, custom models, at a much faster time in much greater numbers. All right, so a little bit of progress so far. Like I said, we're working in the eye models, but at the company, we're already developing these pig models for monogenic liver diseases. We have companies that are involved with us in the heart disease. These are all pharmaceutical companies that are wanting to do clinical trials. My hope is that we're going to add this to the list. So we're working on 2 eye models as of now. One is the Stargardt disease model, and another one is a choroideremia research model. And I already mentioned this to you. Why a pig model? Because the parameters of the pig eye are about the same as a human eye. Very close, right? Don't need to go into all of these details, but you have to trust me on this. We already use corneas from pig for treatments in patients, right? It speaks to itself. So the preclinical CHM model, we argue, will have a better translatability for gene therapy findings to human patients. We believe the surgical mechanisms, the delivery mechanisms would be identical. The similarity in size, again, will allow us to eliminate the guesswork to get into a clinical trial into patients. Couple of scientific slides. Very few. What we're trying to do, we're taking several routes, several shots at the same goal. Ultimately, what we're trying to do is, this is a gene. It's a very long gene, similar to that in the humans. It's coded by 13 exons. I mean, you don't need to go into the details, but we're trying to make mutations so that a functional protein is not made. That will end up causing the disease phenotype. Okay, so then we can start delivering the functional gene, so gene therapy, stem cell therapies, and so forth. We are using this specialty pig model called Osba pigs. So these are black pigs, so they have pigmented retina. That's important for what we are trying to do. So we have this resource at our company that most others don't. So I think as a proof of concept, we initially are done with the first phase of the project. So we already developed a CRE driver that allows us to do an RPE specific knockout. So you have retina, behind that is a pigmental epithelium. So we're trying to make 2 different models. One is an RPE specific knockout, and another one is a retina specific knockout, because there's still some argument as to where the pathophysiology is initiated and where it is more important, right? So like I said, we are developing two models. One is an RPE-specific knockout. This is conducive for stem cell therapies. So this is the epithelium that's behind your retina. And another one is a retinal-specific knockout. So this is where you can just inject your dosage into the eyeball and hope to cure the retina itself. So right now, the pigs are being made. We hope to deliver this to our collaborators who is in Maryland, Dr. Kapil Bharti's group at National Eye Institute. And he will be working, we'll be working closely with him on the cell therapy space. We already forwarded the cells to him to make stem cells from them. So we already started working on them. So idea is that you make the pigs and you already have stem cells from the same pig. So this would be an autologous transplant. So this would be similar to a patient-specific stem cell going back into the patient, right? Just an update. This is how these piglets look like. This is for a Stargardt disease model. We already have those pigs on the ground. They're already shipped to NIH. A special thank you to my collaborator. I mean, my interest in eye research was actually sparked by this guy. Dr. Kapil Bharti is very reputed and very respected scientist in the field, and it's fun to work with him. And as I said here, we're looking for collaborators. Once these pigs are made, once we have validated these models, we would like to get them into as many hands as possible. I mean, we're not limited, right? So, and again, it's easy to do that as a company, because we own everything, right? We don't have the university in the way. The company is at Maryland. We have the facilities to do surgical transplants. We have, as it says, we can house up to 500 pigs. We have the ability to do a very large clinical trial on site itself. And we have the ability to do GLP studies as well, which are important for FDA. So with this, these are the folks who work in the lab. They make all of this happen. These are the two groups, one at Missouri and one at ReOVAte, which is a company in Maryland. And finally, a really special thanks to the Corrademia Research Foundation. I would not have gotten into this field without the generous support from the foundation. So put in a necessary seed funding to get us started on this project. So with that, I'll take any questions you may have.
Yeah, go ahead, please. Yeah, please.
Point was the time it took to develop the pig model. Is that correct?
So the technologies were lagging behind. So we didn't have the engineering tools that are now at our disposal, right? So something as simple as making the genetic modification used to take us a long time. But now with the discovery of CRISPRs and the genome editing technologies, we can now make them happen. So technologies have enabled us to leverage this model more than we could in the past.
And it all makes sense.
Yeah. Yeah. Yeah.
Well, let us get you a microphone real quick. It's coming.
Thanks. So building on the question just asked, I was still wondering, are there any legislations or political boundaries still? I can imagine that getting approval, for instance, now with a pig might be controversial still. So it might be difficult to get something approved. That's question one, and I'll get to the next one.
Actually, it is not, because if you look at all the devices that are going into patients, right, stents, any one of those, they're all developed in pig models. So FDA already recognizes as a good preclinical model. And this may happen pretty soon. This could be a game changer for us. There is a company that is working on a gene therapy application for cardiovascular indication. They are going to FDA and said, we're working with this company. We're going to get all the preclinical data in pigs, and we don't want to do any more research in primates. So, and I would argue that probably they'll weigh the requirements going forward. So you could see that this will become the paradigm going forward. So go
Great.
from cell to pig and then go directly into patients, at least for some indications. I don't want to paint it to the broad brush. Yeah.
Great, thanks. The second question I had is, this morning we saw obviously that there are also some developments in that skin cells can be taken, brought back to stem cells, where a model can be made of a working a photoreceptor, for instance. I do see the difference between the two, but where would you advise, from your professional opinion, when should we work with a pig model, and where is it reasonable to work with a skin cell we retransform to working photoreceptors, for instance? Where lies the balance when we choose one model or the other?
Yeah, so it's a really good question. So what you do is in vitro, we call it, in the dish. You can ask a lot of questions in the dish that you don't have to do it in the animals. Because at the end of the day, we have to follow the reduction. So we don't want to go and make a lot of animals for no good reason. So if a question can be asked in the dish, you ask in the dish. Then you want to test a therapy, we argue that that's not going to get you there. You need to go into an animal for therapies. Make sense to you?
Yeah, absolutely. Especially like long-term effects, for instance, you were presenting, that totally makes sense to do on an animal and not in a dish, for instance. So thanks.
Yeah,
great.
Yeah,
yes.
please, yes.
Hello. I run a gene therapy department in Heidelberg in Germany. And I worry, I mean, I think this is a beautiful, elegant model. I think every single research institute that I know of has an animal facility that can accommodate mice and can work with mice. But even working with mice is prohibitively expensive.
Yes, it is.
We all had access to the CHM mice, but not many of us could afford to use them. And I know this foundation could not afford to allow us to do that. So I worry, even with this amazing model, you will actually limit the number of people who can actually work with it. And I know the researchers in this field, they're decreasing in numbers because of the capacity to do work on this. So I worry that, you know, even with this model, we might be able to do less work with it than we could with others.
Yeah, that is a concern for us too, but I'm more optimistic on this side of the pond, to be quite honest. So we're already talking to a couple of pharmaceutical companies on this space. They're interested in eye models, so they're already engaging the Stargardt disease model, and we hope to talk to them about the CHM model too. Yeah. Yes.
What's the? I'm not asking for a quote here, so you don't have to actually give me a contract. But 500 pigs in your facility running a trial. What's the cost of that? What's the
So it's
1 million, 10 million. What do you need to do?
I'll take your 10 million, but we don't need that much.
Yeah, well, approximately. What's the cost of each rep?
So we can talk offline.
Yeah,
probably that's not good.
He's because he's got a private company. I won't let him answer that question. When I was doing animal research, large animal models, $5,000 a month to house a pig. That's after another procedure. So if you're doing gene therapy, it's probably cheaper than that. It's about $10 a day to house a pig, roughly speaking.
Thank you.
All right. Thank you.
Any other questions? So I have a question. So let's say that we successfully, and then Eric will get you right after mine. Let's say we successfully generate a pig model. We obviously have some investigators lined up, Dr. Barty. Are there any other pharma or investigators lined up that would use a pig model that you're aware of?
So we have some preliminary conversations with Dr. Maureen McCall at University of Louisville. She's interested in gene therapy. She works with other pharmaceutical companies as well. Maybe I don't want to put the cart in front of the horse, but we are engaging a pharmaceutical company right now, and we'd like them to adapt this model.
Thank you. And I would like to say that in the conversations that the CRF has had, one of the things that we made very clear is that if we're going to fund this research, that we want, if someone wants to use the model, they can get access to the model. Now, of course, they have to pay for the model, But there's a lot of companies that will build a model like this, and then actually they'll patent the proprietary. It becomes proprietary and patentable to the organization that developed it, and then no one else can use it. And so one of the things that we've done is said, hey, if we help fund and develop this, we want to make sure that it's commercial available for any academician or investigator that uses it. And we're trying to do it in a way so that if a pharmaceutical company came in, they couldn't then just alter it a little bit and then patent it and then exclude everybody else. So we're trying to be very thoughtful and proactive about that as well.
Yep. All right. Thank you very much.
Am I without my question? Okay.
Eric, please.
Okay. All right. No, just quickly, just so people fully understand this. And I was just at a conference with Kapil Bharti. As a foundation, we definitely were looking at this because when you get into the stem cell therapy, for everybody to understand, we're looking at the transplantation of RPE and photoreceptors.
Mm-hmm.
And to do that, you need a larger model, one for the preclinical understanding of the surgical procedures and things like that. Dr. Bharti is already collaborating with David Gam, who you'll hear a lot more from. And this is helping us get the building blocks necessary that when we get to the point of being able to have a potential therapy, that this model is there so we can speed it along.
Yes.
Because right now, Dr. Bharti is already transplanting RPE, but they're in a porcine model, a pig model that has had its RPE cells killed by laser.
Right.
But if we can have a CHM model that is very true to the CHM phenotype, then it will also help us in better understanding when they do the transplantation to make sure that it is efficacy shown.
Yeah,
Thanks.
yeah. So just real quickly, since you brought up Dr. Bharti, so we already sent a couple of pigs to them, to Dr. Bharti's group. We also, from the same cells we used to make the pigs, we already send those cells as well. So he can actually make stem cells that are autologous to these pigs. So we're already thinking ahead on this line. So, all right, thank you.